I think it started. I didn't see the 3. You know how it normally says 3, 2, 1? It didn't do that, but hopefully we're recording. Yeah, 6 seconds. Okay, we're good. We're golden, pony boy. All right, I'm going to go ahead and start the class. All right, okay, and I went to the next page. So chapter X, no, this is for chapter 16. Sorry, I didn't change that part up there. So this is for chapter 16. And wait a minute. Is this right? Yeah. Yeah, I got it all. Okay, good. So we're going to talk about what is population genetics, what might cause allele and genotype frequencies to change, what is the result of allele and genotype frequencies changing, what are the random events that can cause evolution, and then I can see I didn't finish it or I loaded the wrong one. I hope I didn't. I'm a little bit nervous right now. But also you need to talk about selection pressures and natural selection. I forgot to add that on there. Okay, so what is natural selection and the different pressures, right? Remember there was, can you remember them right now? Let's see if we remember them right now. I put it in the presentation. Directional, how about more? Disruptive and stabilizing and sexual selection. You remember all those? Okay, so I didn't add it here, but I will before I send it to you in Google Classroom. And then don't forget to do your highly suggested reading and thinking. Okay. Can you see the code, those that just walked in? All right. So next, okay, look through your, I'm recording right now for posterity for the kids who can't come, who are at APUsh.
Just so you know, I didn't change any time. I'll fix it on that. There was a typo on the thing.
Okay. 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 Thank you. So make sure that I think the first one, Hardy-Weinberg, we talked about that. You already have all of these. And then I have another one, Introduction to Natural Selection. So just read through your hyperdoc and make sure you feel comfortable with all those. We won't take the time to do that right now. All right. So the big push at the beginning was all population genetics, right? So now we're not looking at the genetics of individuals like we did with Mendel, where we had one cross with another cross, right? Two people and what their offspring. Now we're interbreeding in that whole entire population. And so the first thing we needed to do is be able to mathematically identify allele frequencies, right? And where did we go with that when hi, I'm just recording for my YouTube. Oh, okay, good. Sorry, you're the second visitor that we've had. And so when we measure a change in allele frequencies, what do we know has occurred? Evolution has occurred, right? So the hard part of this was just calculating those frequencies, right? So let's do one practice problem together, and then I'm going to later I gave you a whole bunch of practice problems, more than I've already given you, just a whole website, and then it shows how to work them out. But we'll do one. So if you don't have a calculator, make sure you do, or you can borrow one from me right here, okay? And get a pen and a pencil, and I'll give you time to work one out. So just get ready for that. It's coming. I'm not quite ready for it, but I wanted you to have a second to get ready. Okay, and then come back to me, right? We have talked about it so many times. I'm quite confident you know the difference between microevolution and macroevolution, right? So can microevolution lead to macroevolution? Yes, absolutely. You accrue enough changes, eventually you can't interbreed anymore, right? And at that point, you're a new species. And then in the next chapter, we've had, we talked about the problems with the definition of a species, right? So keep that in mind. But at some point, you're delineating and saying this is a new species, then it is macroevolution. All right. So let's see if we remember the 5 fingers of evolution. These 5 things can cause the allele frequencies to change, which means evolution is occurring. So if we don't want evolution to occur, we have to do the opposite of these 5, right? Does that make sense? Okay. So the pinky, we talked about small populations, and the reason why we were concerned with small populations is they're more susceptible to chance. And what you call that in population genetics is drift, right? Because a small population is more impacted by a 10% loss than a really large population, right? Okay, so we talked about the two forms of genetic drift, the bottleneck effect and the founder's effect. So remember, the bottleneck effect is when you're going through some sort of what? Disaster or crisis, right? That's a bottleneck. And not all members of your population are going to show up on the other side. It's only the survivors that show up on the other side. Okay, that's a bottleneck. And then the second one was founder's effect. And this means I'm starting a population with a small sample from my original population. And it may not be in both bottleneck and founders that this new population that's either getting formed due to a crisis or you're forming a new population, either way, they may not accurately represent the original allele frequencies. And so they may drift towards one allele or another. Okay, so small populations can lead to changes in allele frequency. And then what was the ring finger? Non-random mating, right? Because you're choosing this one is better over that one. And so it's going to increase whatever alleles they're carrying, it's going to increase the frequency of those alleles more if they're more highly prized or valued. Okay, what's the third one? Mutation. This is also chance, right? Genetic drift is chance. You don't know who's going to make it through the disaster, who's going to be able to get on the boat. Mutation is also chance, because they can just randomly, spontaneously make a mistake, and that can cause a change in allele frequency. What's the pointer finger? Gene flow, right? So new alleles coming in, or some alleles leaving and saying, I don't want to live here anymore. And then the thumb is all about selection, right? So selections lead to adaptations. They're both interwoven, right? And so what you're saying is that some traits are better than other traits. So some adaptations are better than other adaptations. So you're selecting for that. So that too can lead to changes in allele frequency. Okay, look at your bio buddy that you're sitting next to today. Are you good? Everybody good? Questions you want to ask me? Yes.
Is founder's effect the one with the disaster? Or is that bottleneck?
Which one is the disaster? Bottleneck. You're going through the neck of a bottle. Not everybody makes it. Founders, you're the founding population, a new population.
Like kind of just decided to migrate somewhere and then start a population there?
Yeah, you could have, let's say, a group of birds that are migrating, and a group of 20 birds get caught up in a storm, and they end up going to a different place, and they set up a population there. Those 20 may not be representative of the original flock of 2,000 as far as allele frequencies go. Does that make sense? They're founding a new population with maybe not the same allele frequencies. Okay, you all right? Okay. So then we had these two equations. So let's talk through these two equations. When we talk allele frequencies, P plus Q equals 1, that assumes if we're not P, what are we? Q. If we're not big T, we're little t. If we're not big R, we're little r. Are there exceptions to that? Yes. So this is an oversimplification. But remember, when we talk about allele frequencies, this is our gene pool. By gene pool, we're putting them all in the same pool, taking our homologous pair, our two alleles, throwing them in the pool together. That's our ingredients that we have to work with in our kitchen, right? And then when we look in that pool, we just count up how many peas are in our pool. Hopefully nobody pees in your pool. Well, that was a little bit funny. How many peas are in your pool? And if you have 1,000 letters in your pool and 300 of them are P, that means that 700 have to be Q because they're either P or Q. That's your gene pool. That's your ingredients. What do you cook? What comes out of your kitchen? You just have to look at the stats, right? If P is 0.3, you agree with that? Then what are the odds I'm going to pull out a 0.3 with this hand? I mean, a P with this hand, 0.3, right? What are the odds that I'm going to pull out another P? 0.3. What is 0.3 times 0.3? 0.09. Use your calculators. Okay, 0.09, and 9 means 9%, right? That's the odd. That I'm going to cook and I'm going to get with those ingredients that I will get a homozygous dominant. Okay? If I'm going to get a heterozygote, with one hand I could pull a P and then a Q, right? Or I could pull a Q and then a P. So that's why we say 2PQ, because there's two different ways. You could go PQ or QP, so we just say 2PQ. So what's 0.3 times 0.7? 0.21 times 2, because it's 2PQ, right? So that's 42, right? So that means 0.42, right? 0.42 is going to be the percentage of heterozygotes, which in a population of 1,000 would be what? 420, right? Okay, Q squared. Those are my homozygous recessives. We said Q was 0.7. 0.7, 0.7. What's 0.7 times 0.7? 0.49. So 490 would be homozygous recessive, right? Yes. Okay, so here, let me write on here. So P, we said P was 0.3, right? Can I write on here? Hey, can I write? Come, look at all my dots slowly appearing. Let's do it. Okay, and then what's Q going to be? I'll just wait for it to appear in like 10 years, right? That's my allele frequency. So this is what my genotypes are going to be coming out of that pool. Who's crawling out of my pool? What are the odds crawling out of my pool that I'm going to get a homozygous dominant? Well, I just have to multiply 0.3 times 0.3. That's P squared. That means the odds that I would get 2 P's out of there, right? So 0.3 times 0.3 is 0.09. So that would be 9% chance that I'll get a homozygous dominant. Right? On Q squared, it's 0.7 times 0.7 is 0.49. So there's a 49% chance I would get a Q squared, which 0.49, right, times 1,000 would be 490. I just did the math in my head. Okay? PQ, 0.3 times 0.7 is 0.21, but it could also be QP, so that's why you multiply it by 2. So that's 42. 0.42 or 420 out of 1,000. Right? If I said not 1,000, if I said 500, it wouldn't be 420, it would be 210, right? You're multiplying it by however many you have in your population. Does that make sense? And it'd be 125 if there were 250. Do you see how I'm doing the math? Okay. All right. So we solved it starting at the pool and working our way out into the population. But you can also start at your population to figure out your pool, right? But who do you pay attention to out in the population? Who's the only one you can pay attention to? Q squared, which are your homozygous recessives. That's the only pure number you have. Because when you look at individuals, both the P squareds and the 2PQs are going to present with the dominant trait. So when you look at somebody with brown eyes, you don't know if they're brown or brown blue. You don't know. So just take all the browns, put them aside, and just focus on your blues, because you know the only way you can be blue is if you're a homozygous recessive. So that's why if in any problem, whether I give it to you or the College Board gives it to you, when they tell you how many in the population have the dominant trait, you want to immediately calculate how many have the recessive trait if you're trying to work your way back to the pool. Because once you have that, all you have to do is take the square root, right? So if they give you a percentage, you would just take the square root of the percentage, right? Does that make sense? Take the square root of the percentage. But if they give you a number, you have to take how many in that population over the total population, right? So if they say, hey, 25% show the recessive trait, then you can go, oh, 25%? Q squared equals 0.25? Okay, then Q equals what? 0.5, right? But if they say 315 show the recessive trait, you're going to be, okay, 315 out of how many, right? If they give you the total number of recessives and the total number that show the dominant, you would add those two numbers together and you would know the total population, right? So you would say 315 divided by 785, whatever the total population was. Now you have the percent, right? And that number you take the square root of. Once you have Q, then you can solve for P, and you can solve for P squareds and 2PQs. Okay, look at your bio buddy and see if they're feeling confident about that. Yes.
So if they say like 25% is the allele frequency, like the recessive allele frequency,
That would be Q.
So then if they say that 25% comes out as recessive, that would be Q squared?
Yes. If you have a trait, you're Q squared. If they say allele frequency, they're talking about Q. Yeah, whenever they say allele frequency, now if they say 75% expressed the dominant allele, that's different, right? 75% express the dominant allele. That's saying 75% of the group has brown eyes because they're expressing the dominant allele. Does that make sense? Then I know, 25% must be expressing the recessive allele, which is 0.25. Now I'm going to take the square root. Okay, what else? You okay on that? All right. So solve this right here. Should I hit pause on the, yeah, you solve it at home. I'm not hitting pause because you solve it at home too. That'd be good for you. So those of you listening at home, it would be, this presentation is going to have a link to a bunch of practice problems. So if this is posted on my YouTube channel, in the descriptor below, I'll put the link to this person has a bunch of practice problems on where you can practice the problems and then go check your answers. So I encourage you to do that. There's a link in this presentation that you may not be able to access if you're just watching this video. And for those of you, if you're my students, I will put it in Google Classroom. Do you guys need a little more time? Raise your hand if you want a little more time. Okay. So if you're bored at home listening and waiting, go down to the descriptor of this video and open up right now while you're waiting, and you can do a few practice problems extra following that link, okay? Until we're ready to go here. Anybody else? Need a little more time? Okay, I'm gonna get going. So what am I gonna work with? Where's my starting point for all this? What do I have to solve for? Q, right? So what's my starting point to solve for Q? How do I solve for Q in this situation? Since I wasn't given the allele frequencies, I was given the population, right? So you're looking at what's come out of the pool. I need to solve for what's in the pool, right? Okay, so what do I start with? Percent that have homozygous recessive, right? That recessive trait. So I know there are 40 white butterflies, and how big is my population? Hi, sweetheart. It's 100. Isn't he getting taller? I think he's getting taller. Wish I was a baller. Okay, sorry. So I would take 40 divided by what? 100. Good. So here I solved it for you. Okay. So I would take 40. Hi, guys. Welcome. I know you were in A push. Come on in. I would take 40 and I would divide it by 100. And when I do that, I get 0.4. So that gives me my Q squared value. Once I have my Q squared value, guys, I need you to not talk when I'm talking, okay? I know you guys just came in for your meeting. Come on in. Once I have my Q squared value, I can take the square root. When I take the square root, and this is where you would screw up if you didn't use your calculator, right? If you didn't use your calculator, you might have gotten a different number there, okay? So when I take the square root, I get Q is 0.63. Once I know that Q is 0.63, then I can solve for P. So the dominant allele is P plus Q equals 1. So P has to be 0.37 because 0.63 plus 0.37 is 1. Do you agree with that? So now I've got P and Q. So the application of that is how many of the brown butterflies would you expect to be able to pass on the white trait? Are the homozygous brown butterflies going to pass on the white trait? No. Who was I trying to find? The number of heterozygotes. Because the heterozygotes are expressing the dominant trait, which is brown, but they are carrying the white trait. So I had to solve for 2PQ. Okay, for those of you that just got in here, here is the code. Oh, I'm scared because it went white. Can you see it? Yes.
So on the question test, do you want us to, if for finding the allele frequencies, do you want us to round to the 100th place?
Yeah, I generally would round to the 100th place. Okay. Yes. Yes, use decimals. They're going to expect you to use decimals, and that's probably how they're going to give it to you on the AP exam. Yeah. So 2PQ, these are brown butterflies carrying the white trait. 2 times 0.37 times 0.63 times I had 100 butterflies. That's what gives me the 47 butterflies. Now, for those of you, and I know you had your meeting for a push for next year, but listen carefully. We went over the expectations of this chapter. One thing I forgot to put on the, I give you the list of what to know. I forgot to mention selection. I just forgot, but natural selection, directional, stabilizing, disruptive, sexual selection, that's all on there. And it's in this presentation. I just forgot to put it on the front page, okay? We went over what population genetics is, the 5 things that could change it. We went through and talked through genetic drift, right? And then we talked through the equations and what the 2 equations mean. So you can watch this, right? I'm going to post it so you can watch this when you get home if you missed that, all right? And then this right here is a link to a whole bunch of practice problems. And if you just go to it, it's a guy's website. Has anybody looked at it? Can you access it? Okay. And if you're at home, I'll put the link in the descriptor of my video. I think he asked like 10 questions or so. And then you click on work them all out. Don't go and look at the answers until you've worked out all 10. Okay. That's the best way. And it gives you really good formative assessment. And then go to the answer and then grade it and see what you got. Right. And if you go into my reviews on my website, there's more practice problems there. Literally Google Hardy-Weinberg problems with answers, okay, questions and answers, and you can get 1,000 questions just to practice that process. But this one was good because he wrote it out like this. I did one similar to this one from one of his problems. It's not exactly it, but you can look through that and then you can see how to do each problem. Okay, you feeling okay about that? Okay, so I'm going to keep going then. So then Hardy-Weinberg says if you can freeze the allele frequencies, you can stop evolution. Why do we even have Hardy-Weinberg? Because that's what people ask me, like, why do we even have that? Yes.
I forget what it's called.
Starts with an N? Null.
Null hypothesis.
Yes. She said null hypothesis, right? We want to see, is there statistically a difference? Mathematically, because you could do chi-square on any of these to see if they're statistically significant, couldn't you? Right? For sure you could. I think I asked you that on one of your Poggles, yes? Yeah, and the one you just did, if you don't see it, I have like meeting after meeting. I have a meeting after this meeting, and then I have open house tonight, but at some point I'll release your Poggles to you so you can look at your answers on that one, okay? But the whole point of it is to show we have to do the opposite of these 5 if we want to freeze those allele frequencies. And if we can't do the opposite of these 5, then evolution is going to occur. So I just put them all on one slide, I think, here for you. Okay, so no mutations, what finger is that? Middle finger. Is that chance? Yeah, mutations are chance. Okay, no migration, no gene flow, what finger is that? Pointer. Good. Large populations, what finger is that? Pinky. And you want large populations in order to avoid genetic, which is more susceptible in a, there you go. Random reproduction, what finger is that? Yeah, your ring finger, because random reproduction is, it doesn't matter. Everybody is equal that you want to have sex with. Okay, in non-random mating, non-random, that's what you want to be, non-random, right? At some future date. Non-random mating is you saying, you know what, you have traits that I like over those traits. So if everybody wants those same traits, then those alleles are going to increase, right? The frequency of the alleles of that person are going to increase. Okay, 5th condition, no selection. What finger is that? Thumb. Can this be the same? Yeah, because sexual selection, really, that's a form of sexual selection. But this is just saying all selection. Nothing can be good. Teeth can't be good. Height can't be good. All of those things. Nobody is better than another. So the conclusion is it's hard to meet all 5 conditions. So therefore, what's going to occur? Evolution, right? Evolution is going to occur. So then let's talk about the thumb. The end of this chapter is just focusing on the thumb and how it can cause differences in allele frequencies. So natural selection says that, first of all, you're going to have overproduction of young, right? Not everybody's going to survive, and there's going to be a struggle. And among those offspring, there's going to be variations, right? And those variations that make you best fit for the environment, those are called what? Adaptations, right? Some adaptations, if they're heritable, if they're in your DNA better than others, over time, those are going to increase and others are going to what? There you go. And that slow accumulation over time, that change in allele frequencies, that's going to be natural selection. Remember, populations evolve, not individuals, right? Populations. So then we looked at different types. Oh no. First, we compared who else said selection, right? Or sorry, erase. Who else said evolution? In fact, who said it first was, but he was off the mark, right? He was wrong, right? Because he had a couple ideas. What were his ideas? Use and disuse and inheritance of acquired characteristics. So if I use it, that my offspring will inherit it, right? If I don't use it, my offspring won't inherit it, right? That was his. So that's not true, right? Darwin would say they're going to be some dead giraffes. Why? Because not everybody's going to make it. But those variations that have the longest necks are going to eat the most food, be the most fit, have the most sex, and produce the most offspring. And over time, giraffe necks are going to get longer and longer and longer. Okay, we good on that? Okay, so we talked about why is there a curve, right? Why is there a curve? Because though we simplified it P and Q, right? Dominant, recessive, That's not really life. Most traits are polygenic, right? There's multiple genes overall that contribute to that trait. So it's not like you say tall, this many were tall and this many were short. There's a whole degree of everything, right? People aren't black and white. We have all different colors that are in here in this room, and I'm glad we do. So that's why you see this curve like this. So the first one is stabilization. Now, in that kind of selection, it's still natural selection, but would it lead to evolution? No, because you're saying the norm is best. Let's not be any different from the norm. So that is a form of selection, but you're selecting for the one that's most common and perpetuating that, and you're staying the course. You're not changing. Okay? Then directional selection, can that lead to evolution? Yes. It's better to be this, be a little more this, be a little more this, and pretty soon on either end of the spectrum, they're not interbreeding anymore. Okay. Now, why is it? The environment could be changing. As you're migrating away from where you started, maybe it's getting a little bit colder, right? Remember when we looked at the yellow-bellied three-toed newt? Remember that? So some of those newts are migrating to where it's colder, so it's better to keep your young inside. Have they stopped mating with the ones that don't? No. But eventually they might, and then you have macroevolution. And that would be a form of directional selection. You're keeping your young in longer and longer and longer and longer. Okay? And then the last one is disruptive. Can that lead to speciation? Yeah. Let's say something changes in your habitat. Let's say there's more trees, and some are adapted to hide in the trees because of their coloration, and others are better out in the open because of their coloration. Then you're going to see those propagate in those two areas, and if they stop interbreeding, then that could lead to speciation. All right. Then the next we talked about, what were the three again? What's this one? Stay the course. Yeah. So that's a form of selection too, right? Because you're looking for it. You might have intra means within. So males fighting with each other, competing, right? If the male is doing the choosing, the head lion, the alpha lion, and he gets to have sex with all the lionesses, right? So that's intrasexual. Intersexual is the females choosing the males, right? We saw that with this jumping spider that grossed some of you out last class. Remember that? So what did he have to do to try to have sex with her? Dance, baby, dance. And it better be colorful and shake around. And whoever can keep dancing and keep jumping, right, gets to have sex and then die. Okay, so that would be Betty choosing Joe. And we said there's two things there. We talked about good gene hypothesis and runaway, of which I think he did both. He had to run away and, okay, anyway. Run away, run away. All right. Then how do we even maintain any variety, right? If there's some that are better than others, we maintain variety in the heterozygote, right? Because the dominant trait can mask a recessive trait. And not only that, sometimes it's better to be the heterozygote, right? Not always. Remember with the dewlap, you either want your dewlap dark or you want your dewlap light, right? It's not good to be the hybrid. But in some case, you actually have hybrid superiority. And we talked about that with the sickle cell. Oh, see how it's still, the heterozygote percentage is still the same. It doesn't matter what extreme we are for Q. The heterozygote number is the same, yes? Okay, so that's stable. But in sickle cell disease, right, if you are normal, so you don't have the sickle cell gene at all, you could die of what? Malaria. But if you're sickle, you're going to die of, but if you're sickle normal, you won't die of sickle cell disease and you're more resistant to malaria. So that's why in areas where malaria is prevalent, not in North America, because we don't have a lot of malaria here, right? But in areas where malaria is prevalent, you're going to see a higher allele frequency of that sickle cell gene because that makes you more fit, right? And it's going to be selected for. Right? In areas like North America, it's going to be selected against. Okay? And then why aren't we perfect? We aren't perfect because life is a series of compromises, right? And so it's a cost benefit analysis. So it's good for that giraffe neck to be longer, and it keeps getting longer, but it started out kind of all tangly around some other organs. So it makes it difficult in its development, right? Or yes, it's good for us to be upright, but as a result, we have bad, yeah, because we didn't start out. We started on the fours, right? Or whatever. Okay. And historical constraints. It would be awesome, right? Less global warming if we could all fly home, right? How awesome would that be? Like have a little wing, okay, and go home. Okay. Yay. That would help us. I would select that. But we don't have that, right? So we are limited by our historical constraints. And then chance is always still a part of it. I know it's covered up. Okay, and I think, yay! Any questions? Okay, I love you. Make good choices.